I would like to thank the organizers for the opportunity to present our work. My name is Tatiana Golovina, and I'm the Senior Director of Cell Therapy Process Development at WuXi. And today I would like to tell you about the innovative closed process CAR T cell therapy platform that we developed at WuXi. WuXi Uptech is a big organization that has capabilities in small molecules, testing, and advanced therapies such as cell and gene therapies. So WuXi Advanced Therapies is the business unit that specializes in development and manufacturing of cell and gene therapies that I will be talking about. Currently, WuXi Advanced Therapies has 4 buildings at Navy Yard, and we provide a range of integrated programs, means that the programs include manufacturing and testing to help our customers take their products from preclinical development to clinical manufacturing to commercialization and then beyond. Our facilities are FDA registered and ICO certified. Currently, we have 4 buildings in Philadelphia, WuXi Navy Yard. So the oldest building is LI1, and this is where we perform the cell and gene clinical manufacturing. So the next building that we put together is CC3, and this is building for late phase and commercial manufacturing of autologous and allogeneic cell therapies. Our third manufacturing building is LI2. This is where we produce late-stage and commercial manufacturing of gene-mediated cell therapies and viral vectors. And our latest addition is R400, which we actually opened this year. And this building is for testing for advanced therapies and standard biologics. And there is a warehouse. And it will allow us to actually increase our testing capacity and move some testing like mycoplasma to Philadelphia area. WuXi objective is to develop platforms in key areas. So therefore, we developed and launched several platforms in recent years in key areas. Thus, in 2017, we launched a platform in lenti-adherent process. Earlier this year, 2020, we launched a V suspension platform, and now in May 2020, we're launching the CAR T closed system platform. So WuXi builds its platform development on its broad experience with CAR T manufacturing. We have been manufacturing CAR T products since 2014, and we have experience in handling lentiviral and retroviral vectors. We do have experience both with autologous and allogeneic cell therapies, and with over 400 released individual patient GMP lots. To be able to achieve this, we built the segregation and chain of identity systems so that we can provide the processing of multiple patients at the same time in dedicated or shared suites. And currently, we add to this capacity the closed CAR T platform. So our work with CAR T platform showed us that the one-for-all process will not work. Therefore, we propose 2 options. The option for the total final product viable cells below 10 billion, we call dose 1, or low to medium. And the strategy for this is one-step expansion. So the second dose 2, is actually gives us above 10 billion total viable cells at harvest. And for this strategy, we use 2-step expansion. Both steps start with the isolation step, for which we use Clinimax Prodigy, CD4, CD8 purification, positive selection, and then we perform the activation and lentiviral transduction, expansion, Then the washing of the final product happens in the closed system, and then we actually formulate and fill in closed system. We utilize off-the-shelf GMP-grade chemically defined media, activators, and cytokines for both CAR-T platform processes. The day zero cell isolation options actually, in fact, include two options. Our preferred option is positive selection, CD4, CD8 positive selection using Clinimax Prodigy. The second option that we can also accommodate is PBMC separation via CPAX. So it can be accommodated based on need. However, restrictions of in-source T-cell percentage and composition may apply here. Okay, isolation efficiency of WBC by Clinimax Prodigy is demonstrated on the graphs to the right. The starting total cell number is between 3 to 10 billion cells, and we get recovery between 20 and 40% of total viable blood cells, with 60% recovery of T cells, and with purity in high 80s, 89 to 98% T cell purity, T cell pure population. On this slide, we present the isolation efficiency of WBC by CPAX FICOL. So it is a bit simpler and more cost efficient than magnetic beads isolation. The WBC recovery is close to the Prodigy isolation, around 60%. However, the average recovery of T cells is closer, it's 56%. However, the purity is variable and between 37 and 63% here. So the next step is expansion using GREX 100 MSCs. And as you can see, there is a plateau actually around day 9, 10, and therefore it actually distinguishes the duration of the process, which is now 10 days. So the viability at the end of expansion is above 85%. So, and the platform can actually utilize the expansion of multiple GREXs. The yield per GREX is 2 to 3 total billion of total viable cells. The final cell population is concentrated and washed in closed system, followed by resuspension in pre-formulated buffer. So achievable recovery is actually in the 90s and above 70%. Final viability is also pretty good in the 90s and above 70%. The final volume is between 100 and 1200 milliliters, with cell densities between 2 to 100 million cells per milliliter. The closed system fill is completed using CPAX with final product fill in pre-evaluated cryobags. Post-thaw, the platform yields more than 70% recovery of cells with more than 70% viability. So here are some observations with the platform process. The CD4 to CD8 ratio is donor dependent and is in the range from 0.31 to 2.6. And the naive and central memory T cells percentage in final product is a bit variable and donor dependent and is in the range between 40 and 60%. We optimized transduction using the CAR GFP lentivector. So we optimized the timing for adding the lentivector to the post-stimulation. So our transduction efficiency may depend on vector and also on the cell's donor. Therefore, we tested that on multiple donors. and our target range is between 2 to 50 CAR-T positive cells. Utilizing our platform allows a speedy pathway to manufacturing by skipping both tech transfer step from the client process to WuXi process development, and from WuXi process development to WuXi manufacturing. In fact, we have all materials in stock, batch records handy, we have qualified facility and trained personnel. So based on that, the customers can save months and months of their timelines if they use the closed CAR-T platform. Here's the outline of the typical suite for closed process CAR-T. And as you can see, the kind of typical equipment includes Clinimax Prodigy, CO2 incubators, Gather Access, Lovo, CPAX, Zuri W25s, tube sealers and welders, and the cell counting piece and C200. WuXi CAR-T platform is an integrated program. Therefore, we also propose assays for characterization and release. At the left panel, you can see the product-specific release tests that we propose, such as CAR expression by flow cytometry and integrated copy numbers. Both are gene of interest specific. We also provide general characteristics such as cell count and viability by NC200, identity by flow cytometry, cellular impurities by flow cytometry, and also safety testing such as sterility, endotoxin, mycoplasma, and replication competent virus. We also offer additional optional capabilities such as, for instance, potency assay, product-specific, phenotyping by flow cytometry, such as T-cell memory phenotype, T-cell activation phenotype, T-cell expansion, T-cell exhaustion phenotype. We also offer in vitro and PCR-based adventitious agents testing and residual viral DNA. At WuXi, we offer technologies that really meet our client needs, such as, for instance, multiple flow cytometry platforms. We utilize BD FACS-Canto for our GMP release testing, and it's industry standard. And we also offer the high-throughput semi-automated flow cytometry for R&D release. So we can screen conditions at high speed and reduced cost. We also offer the ddPCR for determining the integrated copy number, which is reliable and no reference needed standard method. We also offer analytical development, gene of interest specific ddPCR method for each client. We offer the stability program with integrated testing. So at the top panel of the table, you can see the tentative time points that we offer. It can be also based on the client request. So the stability studies can be initiated at any products, on any products during clinical manufacturing. So the protocol include a measure of product sterility, identity, purity, quality, and potency as recommended by FDA. So, and also intermediate stability studies can be performed to evaluate process hold times, stability during cryopreservation, and so on. WuXi also offers various temperature conditions to help assess the product stability. WuXi has a long history of regulatory compliance, and during the last 12 months, we were audited by 4 major world regulatory agencies, and we had very good results. So we also offer the regulatory affairs program, which is, for instance, IND and BLA authoring and submission, CMC facility and material controls. We offer DMF authoring and submission, RMIT and PRIME designations, briefing documents creation, annual product reviews, PI inspection, readiness, and marketing authorization support and maintenance. Overall, WuXi Advanced Therapies platform advantages is to accelerate progress and time to market with greater predictability. So we have established equipment and process in stock raw materials with batch records. We have GMP facilities with dedicated and shared suites and established and dedicated teams in process development, analytical development, quality assurance, and regulatory. We also have the manufacturing process and integrated analytics, strong and dedicated technical guidance, including regulatory support, and fully integrated in process and release testing for greater predictability. And with that, I would like to thank the audience for listening to this presentation. I would also like to thank WuXi leadership for support, as well as WuXi technical teams and launch teams for their hard work and dedication. Thank you.